Can you hear me? Oh, it's a relief to take the mask off. Is there a pointer, or should I just use my cursor? Let's see, it must be a pointer. I don't...
You're on a budget.
Good Lord, I didn't know it was that bad. Okay. Let's see. Thank you. Okay, ready. I'm honored, I really am. It's been a long time at Berkeley. As you'll see at the end, over 4 decades. I was tempted to leave, maybe once or twice, but never could pull myself away. So thank you for the invitation. It's really wonderful. Also, I am thrilled to be standing here without a mask and looking at real people. I have been completely on Zoom, pretty much, for the last 2 years. So hopefully I haven't lost the skills of standing here and operating non-remotely. As my title implied, we're going to be talking about an interface, and it's an interface that has fascinated me for a long time because I'm trained classically, and what chemists look at is reactions that go over the top of barriers. But quite a while ago, we started making observations that suggested we needed to think quite differently in terms of the quantum aspects of reactions, in particular, moving through the barrier rather than over the top of the barrier. So here we are, connecting physics and chemistry. I'm trained as a physical organic chemist. I was initially in the Department of Biophysics at Penn, but I always thought of myself as a biochemist or even an organic chemist, to the horror of the organic chemists perhaps in the audience. But actually, I don't know where I fit in chemistry, and it's at the interface that some of the most exciting work gets done, in my view. So let's look at some connections and legacies. Now, I was reading about G.N. Lewis in preparation for this lecture and discovered some things about him that I probably don't want to talk about here, but the important connection that was compelling has to do with deuterium, because I'm going to be talking about isotopes a lot today, and have only recently recognized how important deuterium was in the career of Mildred Cohn, who was a professor at the University of Pennsylvania, who was a colleague, a friend, and also an inspiration for me. And so the lineage is, G.N. Lewis, of course, was the PhD advisor of Harold Urey, who discovered deuterium, who then was a PhD advisor for Mildred Cohn, who was the first female graduate student in Urey's lab at Columbia, and possibly in the entire Department of Chemistry, who later became my friend and inspiration. So science is a very small world, and the idea that it would be deuterium that would link us all is really quite fascinating to me, anyway. So basically, we decided many years ago to study CH activation. It's a very important area in chemistry. And of course, enzymes are functioning, at least primarily today, in aerobic environments. And the abstraction of hydrogen from carbon provides a lot of the energy that we need for life as we know it. Now, trained as a physical organic chemist, I started to look at transition state structures. And we could do that either by looking at structure-function relationships or looking at kinetic isotope effects. And back in the 1980s, we made an observation, which I remember speaking about at Gordon conferences that no one could explain to me at the time, which is that the transition state structures that we inferred from these 2 physical organic probes came up with a different conclusion. And of course, these were just the standard protocols in textbooks in terms of how you could study transition states, which is what reaction kinetics was all about. After coming to Berkeley, this was published, the work had been performed prior to coming to Berkeley. After coming to Berkeley, we started to look at the relationship of protium, deuterium, and tritium. And another interesting anecdote, Mildred Cohen had said to me she wanted to use that comparative study herself, but tritium was not available to her when she was a graduate student in Yuri's lab. And I didn't realize that until after the fact. Sometimes we absorb information and we're not even aware that we're absorbing it. In any case, we looked at H, D, and T, and what we found was non-classical relationships. Now, the classical properties of the isotopes of hydrogen are conceived in the context of zero-point energies and the dependence of the zero-point energy on the mass of the particle. And we found relationships that led at that time to the proposal of tunneling in enzyme reactions. So this is the first publication where we come out publicly and claim that the way hydrogen is transferred is non-classically and through the barrier. Now, as time went on, we looked at many different systems. Eventually, we started to pick up reactions that showed isotope effects that were huge. This is all at room temperature. In fact, 100 is no longer considered unusual at room temperature, whereas in the textbooks, the maximum is 7 to 10. And later, we came up with isotope effects of 700 at room temperature. And this has to do with the different tunneling properties of H and D. Finally, there's this very curious phenomenon that we call temperature-independent KIEs, which cannot be explained classically either. Okay. Now, everyone knows that electrons tunnel, and the wavelength of the electron is long enough that you can have the expectation of tunneling over 10 or 20 angstroms. Since the wavelength of a particle can be estimated from the de Broglie wavelength, you can take a look at what the wavelengths of protium, deuterium, or tritium might be in the range of 0.4 to 0.7 angstroms. And because of the 2,000-fold difference in mass, you might say, well, why would hydrogen tunnel at room temperature and the electron, well, easily explained, it's not so obvious for hydrogen. But the key feature is the process that you're looking at. If the wavelength of the particle is similar to the reaction path, then it will act as a wave. And many hydrogen transfer reactions are formalized as occurring over a distance of 0.5 to 1 angstrom. It's bingo, right in that sweet spot. And in fact, it's not unusual at all that hydrogen will behave non-classically. Now, one value of looking at tunneling using hydrogen rather than the electron is that we have isotopes. And they're going to allow a much more clear-cut demonstration and understanding of the implications of tunneling. And what I want you to note is deuterium has a shorter wavelength than protium. And if things get out of sync and no longer optimal for through-the-barrier processes, deuterium's going to have a hard time. And that shows up in some of our studies. So here's the door opening. Let's look at biology in a different way. Okay. Enzymes. I'm extremely stubborn scientist, I must say. I've been trying to figure out enzymes for the entire career. And the one factor I want you to realize is that enzymes catalyze rates as much as 10 to the 30-fold over that of the uncatalyzed reaction. This is not an exaggeration, it's a remarkable number. I would say at this time we still lack a satisfactory and predictive set of principles that underlie these extraordinary rate accelerations. I mean, we understand structure function in DNA and other macromolecules, and yet we still lack that set of principles that we could go into the laboratory and de novo design an enzyme. So let's see where we can go. Okay, this just gives you an idea. There's been a lot of interest in computer design. And the tan curve shows you the average rate acceleration that computer design enzymes are able to achieve. It's in the range of about 10 to the 7. Here is the red bar represents the distribution of the rate accelerations by native enzymes. Of course, directed evolution is a powerful tool. You can de novo design a scaffold that may begin to catalyze reaction and then subject it to directed evolution. And what you see is a large increase relative to the non-optimized situation, but it's still far away from the native enzyme. Okay, what's going on here? Well, what chemists do, or have done, is look at the active site of proteins. They're well defined. We have hundreds of thousands of structures to look at. We can understand the metal ions, the organic cofactors, et cetera. And the scaffold has been referred to as just that. It's the framework that holds all the active site components into place, enabling them to interact with each other and provide catalysis. What about the scaffold? And so I say to myself, it's the scaffold, stupid. You know, there's something that we have been ignoring. Now, of course, proteins are large, active sites are small, and they're moving all the time. They're jiggling and wiggling constantly, and over wide differences in, wide changes in angstrom configurations and different time scales. So we have large change conformational changes, domain shifts, loop closures, and then the more subtle movements. So the kinds of changes in structure can vary anywhere from 5 to 10 angstroms to hundreds of angstroms, and the time scales can be anywhere from seconds to femtoseconds. Oh, how interesting that is. I'm just warming up here. Let me just see. Okay. Huh. They would like to record this. Deny. Sorry for that. I was there. This is not a photograph. This is a picture I took of a tiger. Okay. Anyway, timescales are very important. So 2 frames of reference. Catalysis is millisecond. In nature, virtually all of the reactions catalyzed by enzymes have converged into the millisecond time scale. Barrier crossings are femtoseconds. What's happening in between? Okay, so what we're going to do is look at the process called proton-coupled electron transfer. This is fundamental to biology. And of course, a lot of the molecular machines that catalyze key processes such as photosynthetic water splitting, oxygen reduction to produce ATP and the energy components of the cell, conversion of nitrogen to ammonia, et cetera, are multiple proton and electron transfer processes. Well, we were very lucky a number of years ago to start to study an enzyme that can be a paradigm for a coupled proton and electron transfer, where it happens in a single active site, but displays the kinds of properties that are expected for the more complicated systems. And Lepoxygenase was the first enzyme to show these extremely elevated kinetic isotope effects and also non-classical temperature dependencies, and led to a very intense amount of activity by theoreticians trying to understand what in the world is going on. So here's a study of Lepoxygenase. The enzyme is a non-heme iron protein. It works on a long-chain fatty acid. It abstracts a hydrogen from the carbon 11 of the long-chain fatty acid, transferring a proton to the hydroxide and electron to the iron, reducing ferric hydroxide to ferrous water. Straightforward, except that the proton and electron start at the same place but end up in different places. Now, this reaction occurs in a buried active site. Here's the ferric hydroxide. This is the interior of the protein that has been modeled with the fatty acid side chain. And the reactive carbon is shown here in black. And what you should note are the number of hydrophobic residues illustrated. This is an extremely hydrophobic pocket, and the substrate is surrounded by these bulky aliphatics. Now, we've been very interested in doing mutagenesis that is non-traditional, which means to take a hydrophobic side chain and convert it not to change the charge or hydrogen bonding property, but just reduce the size and look at the impact as we introduce impairment in packing defects within the protein and ask the question, okay, we're slightly perturbing the scaffold. How does this show up catalytically? So here we're looking at one side chain which is a helix turn away from the 5-4-6, which sits right behind the carbon 11 that's undergoing hydrogen atom loss. And we simply introduced a series of other aliphatic side chains. You can do this. You don't need unnatural amino acids. You can vary shape and size. And what we saw was that, first of all, the enthalpies of activation, these reactions occur at maybe 300 per second with small enthalpies of activation, very unusual. But the most unusual property was, and we knew the chemistry was rate limiting, hydrogen transfer is rate limiting, and we measured the temperature dependence to get the enthalpies of activation for protium and deuterium transfer, and we saw this trend. And all we did was reduce the size of a side chain, one helix turn away, and all of a sudden the energy of activation for D transfer went up disproportionate to the energy of activation for H transfer. So here we are running into this situation where deuterium has met a wall, if you will, and you need a significantly higher energy of activation to transfer deuterium. Now let me just say that the temperature dependence of KIEs is part of physical organic chemistry. I don't want to belabor the point except to say that there are different shapes if we use protium or deuterium, and this is attributed to different degrees of tunneling at different temperatures. And hydrogen tunnels at lower temperatures, and deuterium tunnels less at lower temperatures. I'm sorry, hydrogen tunnels more at lower temperatures and then continues to tunnel as you go to high temperature. And the increase in, the decrease in tunneling occurs faster with deuterium than protium. Okay, but the main point is we have three regions. One where the isotope effect looks temperature independent, another one where these two curves converge at the same point, and a third intervening region where the dependence of D is sharper than the dependence of H on temperature. Three regimes. At temperatures that go from absolute zero to infinite temperature. However, enzymes show the same temperature near room temperature, the same property near room temperature. So here we see behavior where we have identical slopes at room temperature. We see plots where the lines converge at room temperature, and then they cross. And all we've done is change a single hydrophobic side chain to a smaller size. Okay. Here is the model that has emerged. This is called a vibronically non-adiabatic model for hydrogen tunneling, and it has a number of features that I want to illustrate first with cartoons, and then we'll look at the equation, but not dwell on the math so much as the physical properties that we're learning about the enzyme. Okay, enzyme substrates are constantly in interchange between active and inactive states. And also there's this wiggling and jiggling of the protein that leads to conformational sampling and leads to somewhat higher energy states. Now the issue for tunneling is that we have to be able to impart energy into the system so that we can transiently reach equal energies for the reactant and product wells. So we need transient degeneracy between reactant and product wells for efficient wave function overlap. And that's the activation barrier for this reaction. So in fact, we have then an additional input which leads to the conversion of the reactant into the product well. Now all of these properties are temperature dependent but independent of the isotope that's put into the reactive position of substrate. Okay, the next coordinate is simply the wave function overlap, and that's shown in D. And this is dependent on the mass of the particle, the distance that it transfers, but it's temperature independent. So what we have are different coordinates for temperature dependence and isotope dependence, and bingo, there's our temperature independent KIE. It just follows beautifully. And all of the theoretical treatments that involve these vibronically non-adiabatic models estimate that the donor-acceptor distance is 2.7 to 2.8 angstroms at this point. Now, how do we get those crossing lines? And in fact, this is a result of the internal defect introduced, for example, by single mutants, where all of a sudden the energy of activation of deuterium has gone up. And this is because you make this perturbation, and now the donor and acceptor can't get close enough to allow essentially equally probable wave function overlap for H and D, and you have to introduce a new coordinate, which is called the donor-acceptor distance sampling mode. So that will explain the trends, but of course we're much more interested in how native proteins function, not just the mutants. And so we're left with this dilemma of these very short distances to get these parallel lines and essentially temperature-independent isotope effects. Here is the math. It's very simple. It follows from electron tunneling theory. And there are a couple of terms that most of you will recognize. Of course, electron transfer, according to Marcus theory, is dependent on the delta G naught, the driving force, as well as the environmental reorganization, which is lambda. This is the exponential term. It's temperature dependent. However, hydrogen tunneling is different because we have, in addition to these classic Marcus terms for tunneling, we have what's called the vibronic overlap, which describes the electronic coupling between the reactant and product, but also the wave function overlap for the proton that is being transferred. And this is dependent on the distance between the donor and acceptor. I already alluded to that. So we have now this additional donor-acceptor distance parameter that's quite important. And then we have one other feature that appeared, and this is a calculation by Sharon Hammett-Schiffer, a longtime collaborator, at a Faraday discussion in 2016. And what Sharon pointed out is that she can explain the experimental observation, she can calculate the tunneling rate, but the observed rate by the enzyme is much, much slower than the calculated tunneling rate. So what was missing? Well, it's because, in fact, the tunneling is a very rare event in the enzyme. We're talking about rare processes. And so she introduced this term, F-conformation. Now, F-conformation can mean many things. It could be defined as a fraction of enzyme that's active to do barrier crossing over the total, or it could just be the probability that tunneling can occur. But this emphasizes that it's such a rare event. Okay, now Brian Hoffman, who is an indoor spectroscopist at Northwestern, is a collaborator. And we went to Brian a number of years ago, about 5 or 6 years ago, to say, can he help us? Because we didn't have a structure for SLO with bound substrate. And we knew that so much of our interpretation depended on the specifics of the arrangement of the substrate in the active site. And we needed an alternate probe to get the donor-acceptor distance in the resting form of the enzyme. It's possible that the protein could generate a very compact active site, and in fact, the ground state distance would be 2.7 angstroms. This would be a kind of rack mechanism for enzymes. And so what Adam Offenbacher, the postdoc in the lab, did was synthesize substrate containing carbon-13, and we proceeded to do nuclear electron spin measurements to get the distance between the donor acceptor and the resting state of the enzyme. Now, this is a detailed experiment that I'll summarize here. Essentially, we could use ENDOR to look at the distance between carbon-11 and which is the carbon hydrogen. This is the hydrogen donor, and the oxygen, the hydrogen acceptor. And so we could measure that distance, and it's shown here for one of the conformers that we detected as 4.9 angstroms. We also had crystal structures. In this case, we're using manganese-substituted enzyme because it had the relaxation properties that allowed the measurement. So we know the manganese-oxygen distance, and through the law of cosines, we could actually calculate the donor-acceptor distance as being at van der Waals. This really ruled out the possibility of the enzyme acting as a rack. And at the same time, we noted that a very large fraction of the bound substrate wasn't even at van der Waals distance, but was out at 3.8 angstroms. So we have all these varying distances. Obviously, the substrate is sampling many conformations in the ground state, and the degree to which we see the active versus the inactive or non-van der Waals distance varies with mutation. So you can go back and do that measurement. with this series that I showed you kinetically, and you can measure the fraction of enzyme that's in close distance, which we define now as van der Waals, and then that fraction goes down as the residue gets smaller. And we can go back to the vibronically non-adiabatic models and actually calculate what the distance is between the donor acceptor prior to the donor acceptor distance sampling. And once again, we get close to 2.7 for the wild type enzyme, and then much longer for these mutants. So things are moving apart, and the ground state is not at 3.1 angstroms, according to this donor acceptor distance sampling mode. This is, in fact, after the enzyme has already activated itself and prior to donor acceptor distance sampling. So we still have this issue. We start out, we know the ground state is 3.1. We have to get it to 2.7 in order to get this temperature-independent KIE verified by these experimental Endor measurements. Okay, so how does the scaffold do this? Now, researchers have been trying for the longest time to connect protein dynamics to function. I mean, it's an extremely active field and quite contentious because there are many people who claim that dynamics aren't important at all. Okay, so we need some new methodology. And what we have done is a number of things. First of all, we've introduced techniques for looking at spatial resolution of dynamics in proteins. Computer science allows us to compute protein networks, but we are experimentalists, and we believe that unless we can measure something, we're reluctant to believe it. Proteins are moving all the time, as I said. They're opening and closing, and when they do this motional behavior, they open up to the solvent. If you place a protein in D2O, the backbone ami can undergo exchange with deuterium, and so you get progressive increase in the deuterium content of the protein. This is a very common methodology in protein biochemistry, in fact. And you can do this under a condition where the protein is opening and closing faster than it's incorporating deuterium into the backbone. So this is the expression, the generic expression, and under this EX2 exchange, where closing is much faster than the subsequent incorporation of deuterium, you get the reduced equation where the time rate constant for the incorporation of deuteron into the protein backbone, here are some illustrative examples, contains an equilibrium constant for the opening and closing, and the kinetic constant for the exchange. And in fact, then, we can say that the energy of activation, which is approximated by delta H observed, is equal to the sum of both a thermodynamic and a kinetic term. That's how we look at the temperature dependence, then, of HDX, rather than just focusing on a single temperature. So we're going to have to understand these two components, and if we want to look at protein dynamics, what we really want to do is separate out the enthalpy term for just the local opening and closing of the protein, which is going to relate to the kinetic behavior. So the next layer of complexity is to introduce a functionally impactful mutation. And we would do that at a single site, and the assumption is that that's not going to change the kinetic barrier for HDX exchange, except at the position where the mutation is. And if we're looking at mutants all the way around the protein, let me just clarify one point, which is that the analysis is not done on the entire protein, but what we do is we quench these reactions by going to low temperature and low pH, break the protein up into small peptides, and then analyze each of the peptides, which allows us to get spatial resolution. So in this case, what Adam did was he analyzed the digested protein, getting out a total of 49 non-overlapping peptides with very good sequence coverage, and he was able to look at each peptide as a function of time temperature, as I showed you on the last slide, but also these functionally impactful mutations. And when you compare the mutation to the wild type, the kinetic term drops out, it's the same, and we're just left with the impact of the mutation on the flexibility of the protein scaffold. Okay, so here's the result. The initial study used wild-type enzyme and 2 mutants one in a series I've discussed a number of times thus far, and another residue which sits behind the reactive carbon of the bound linoleic acid. And the energy of activation for catalysis is shown on the right, and some of the variants, one variant, it goes down, the other variant, it goes up. And when we look at the energy of activation for this exchange of deuterium from the solvent into the protein backbone, similarly, the I553G shows a reduction in HDX, and the 546A shows an increase in HDX. We're seeing a correlation, just a correlation so far, between the energy of activation for the hydrogen deuterium exchange that reflects changes in protein flexibility with mutation, and the energy of activation for kcat. This is shown in only 2 peptides out of the 49 analyzed, and it's in this orange box and then blown up, where you can see now there's this region of the protein which site-specifically is impacted by site-specific mutagenesis and gives trends in energies of activation for changes in flexibility that map to the energy of activation for changes in kcat. If we then extend this study to include other residues in this, this network, as well as a second site represented by L754, which is off the network, and as shown in HDX, when mutated to alanine, does not change the energy of activation for hydrogen deuterium exchange. In this way, we can distinguish residues that are what we say are on the network, are sensitive to catalytically impactful mutations through their changes in flexibility, from those which are indifferent to the mutations that affect catalytic activity. All right, so we have on-network residues and off-network residues. And this is a picture that will summarize where the thermal network is located in the protein, which takes into account the HDX exchange data together with the X-ray structure, where we can actually map a conduit that leads from the active site to the solvent. Well, we were not satisfied that we had defined this problem completely, and therefore turned to a second experimental tool, which is called room temperature X-ray analyses, in which mutations once again are introduced into the protein. And now what we're looking at is the impact of mutation on the conformations of protein side chains. And once again, what we see is when we make these activity impactful mutants, the side chains that are altered are restricted to this region that we've identified as the thermal network, but extend the region of the thermal network into a somewhat expanded shape. And what I'm just showing you are the electron density maps that allow us to identify which side chains are susceptible to mutation via room temperature X-ray crystallography to supplement the information from the hydrogen deuterium exchange. So here is the result for soybean Lepoxygenase. What we have is a very select cone of residues that lead from the active site into the solvent. And in fact, we can show through this distance map of contacts that these are all interconnected through van der Waals contacts and lead over 20 to 30 angstroms from the active site iron out to the solvent. This is really quite unique. This is really a first of its kind, where we've taken these different biophysical probes and probes of kinetic activity and been able to site-specifically define a thermal network. Okay. So this is a spatial resolution. What about a temporal resolution of protein dynamics? What I've shown you is a time-averaged analysis that is very much analogous to X-ray crystallography, in that we really haven't addressed this time regime between femtosecond and millisecond. Okay, now the unique feature of this hydrogen deuterium exchange exposed thermal network is that it terminates at the solvent interface. And of course, many investigators have been interested in the role of water as the thermal bath and the origin of thermal activation of proteins. And this is a very nice paper by Bakshi, where he distinguishes bulk water from biological water. And the relaxation rates of these two classes of water are quite different. The bulk water relaxes in the picosecond regime, and biological water in the nanosecond regime. And so really what we wanted to do was look at the protein-water interface now and try to analyze the time constants for the activation of this network and relate it back to the femtosecond barrier crossing. Okay. So what we're going to do now is we install an organic probe onto various loops. This is the full picture of the SLO structure, which includes the loop that we have identified at the end of the hydrogen deuterium exchange and room temperature X-ray exposed activation loop. That's called Q322. We place a cysteine into this position, as well as we install a cysteine into another loop on the other side of the protein, which is a control loop. And we then label these cysteines with a fluorophore called bedan. And this is a very beautiful protein chemistry done by Paolo Zarazoga, who is a postdoc in the lab currently. And of course, bedan has been used in proteins to look at motional events. But what we're doing is looking at the motion of a probe that's out on the solvent and 30 angstroms, 20 to 30 angstroms away from the active site, and trying to understand its, the time of its relaxation and also the temperature dependence. Now, in this case, we're looking at Stokes shifts. And the Stokes shift is a measurement of the relaxation of the photoexcited dipole in the fluorophore by the solvent. And we're studying this as a function of time and, importantly, temperature. And of course, at this point, we have the comparison of the probe, which is in the experimental thermal network, 322, and the control loop. And we're seeing relaxation rates that are in the range of 1 to 10 nanoseconds. Now, the feature that we were most interested in was the temperature dependence of this relaxation. And what I contrasted here is the control loop with the experimental loop. And in fact, the control loop shows an energy of activation that is essentially 0. And the catalysis energy of activation is 2. Whereas the bedan at 322 has energies of activation which are matching now the energies of activation for catalysis. This is only one type of measurement, but suggested to us that in fact, the relaxation rate at the surface of the protein, which is reflected in the Stokes shift's behavior of bedan at position 322 and is occurring on a nanosecond time regime, is in fact equal to the energy of activation at catalysis. And this was a result which in fact suggested a cooperative protein motion that was controlling the activation of enzyme tunneling, and that originated at the solvent interface. Okay, now this is a very provocative idea and certainly not previously demonstrated. And what Paolo proceeded to do was extend the study by making a series of mutants at the active site, again, and looking at the impact of those mutants on the relaxation rates for the bedan, which is located on the protein surface. And what I show you here is a plot of the energies of activation for the slower relaxation rate, which is normally attributed to bound water in proteins, and the energy of activation for catalysis. And so we see the wild type and then these 553 variants that we've talked about previously, another variant which is further out toward the protein-solvent interface, and then the residue 546 that sits behind the reactive carbon-bound substrate. And lo and behold, we see a straight line of 1. Now, what that means is that the energy of activation for the relaxation of the bedan, which is now located at the protein-solvent interface and which has been looked at via molecular dynamics simulations and does not appear to be folding back into the interior of the protein, is showing the same energy as activation as what we're seeing for catalysis. All right. So this to us is very strong evidence, it's not a proof, for a network that in real time is giving us motional effects that serve as the thermal push to achieve the tunneling properties of the reaction coordinate. If we look at our control 754A, which is off the thermal network, we don't see a correlation at all until we introduce a second site of mutation, which is similar to the mutations that lie along the red line. So we get a shift, a parallel line, but we do not any longer have a one-to-one correspondence between these two energies of activation. So my question to you in the audience now, having led you through these spatial and temporal resolutions, is how do you explain this phenomenon? Okay? Here it is, and this is what I think is going on in proteins. We're going to explore this a little further, but essentially what we have are cones or regions of protein which, by virtue of the anisotropy of the protein itself and the exposure of the exterior of the protein to the bulk solvent, undergoing a cooperative thermal activation. We call this a protein quake, a thermal protein quake. It's cooperative, it's rapid, and it's transmitting thermal energy from out here, protein solvent interface, into the active site. Now, how does this relate to the bulk solvent? Well, the bulk solvent is, as I said, the heat bath, and what we have is a region of protein that is particularly susceptible to heat flow as a result of the collisions with the bulk solvent. And heat flow through proteins is a well-studied phenomenon and can easily occur over the 20 to 30 angstrom distance between the solvent and the active site on the time scale of picoseconds. Okay, so what we have is picosecond continuous activation, flow of heat in and out, and every now and then, the heat that reaches the active site can generate a configuration that will lead to catalysis. Catalysis, once again, is a rare event. And so you have then this whole series of potential rate constants with similar activation energies. The Stokes shifts themselves show a nanosecond time constant. Well, you can imagine that the number of interactions that will allow solvent relaxation of an excited state dipole are much smaller than the number of exquisite interactions that have to occur simultaneously in an enzyme active site for catalysis to proceed. So the probability of catalysis is very low. It's a very rapid event, but the probability is low. And then as we move down this cone, we have different biophysical measurements with different probabilities of occurring, but they're all linked to this common thermal quake. Now, I just want to show you a molecular mechanism for how then to relate the classical scaffold to the chemistry at the active site. And here is that cone again that I've shown you as a result of HDX and room temperature X-ray studies. Here's the active site iron. Here's the bound linoleic acid. And here is 546 that is at the tip of the thermal network and is in van der Waals distance with the reactive carbon to substrate, which is within van der Waals distance from Endor measurements of the hydroxide where it's going, of the iron, where it's going to transfer a proton and electron. And this is the off-network residue 754 as a frame of reference. Now, if we look at this more carefully, what we see is that a remote carbon on the linoleic side chain, whether we model it in or out, is within van der Waals distance of the C-terminal carboxylate, which is a ligand to the iron, and is going to regulate the electrostatics at the iron, which is, of course, going to regulate the ability to transiently get degenerate energy levels between the reactants and products. So we have now the C11, which has to get closer by 0.7 angstroms to the hydroxide, and now we have a hydrophobic residue near a carboxylate, which, as it moves, will regulate the electrostatic charge, distribution, the ability of this carboxylate to hydrogen bond to the adjacent oxygen, and overall the electrostatics at the iron, which allow this transient degeneracy. This is really an orchestration. This is the conductor with his baton allowing to flow transiently in and out, reaching the bound substrate at two key points, the reactive carbon and a remote carbon, which are simultaneously altering the two parameters that control active site tunneling, the distance between the donor acceptor and the electrostatic energies between reactant and product, which are generally not equal to 1. In fact, the reaction is quite exergonic. So I think on a molecular basis, it works. Now, I am running out of, I'm sure I'm over time. Am I over time? I just have so much to say. Can I keep on going? People are going to stay. Good. What time?
It's a little after noon, 12.
Okay, we have another 10 minutes, I would think, easily. Over 15. All right, 15 even. All right, stay put. Stay put. Okay. So I want to talk, so the question is, are the data for SLO a coincidence? When I show these data to people, they say, oh, it's just a coincidence. I don't believe in coincidence. We're scientists. We don't believe in coincidence. When we see something that we can't explain, we really need to step back and say, just like with the tunneling, yes, it can be non-classical. Now we're seeing behavior that leads us to another kind of dark alley where, oh, it's just coincidence. So the second system I want to tell you about is a different hydrogen transfer system, a different mechanism. It's a hydride transfer. It uses a cofactor and not a metal. And it was started a number of years ago where we see, in fact, very unusual rate and kinetic isotope effects as a function of temperature. And we see both temperature-independent and temperature-dependent isotope effects in the same system. And early on, when we were first exploring HDX exchange with my former student and colleague and collaborator, we tried the HDX measurements just at a functional temperature and a non-functional temperature. So we looked at HDX as a function of the break and behavior. And once again, we were able to define a region of the protein now that communicated with the solvent interface and reached into the active site. Here is the iron. Substrate binds in this region and cofactor here. Of course, we didn't fully understand what we were seeing at the time, but looking back, it's the same similar result where we have a very site-selective region of the protein that changes its properties in hydrogen-deuterium exchange as we impact catalysis, in this case, just by changing temperature. So what about the temporal aspects? We now have this spatially defined, thermally averaged region that we're interested in. What about the temporal aspects? And so now we're not going to look at the protein-solvent interface. We're going to use the 3 internal tryptophans that are known to be present in the native enzyme. And we're going to compare by making single tryptophan variants, which are quite functional, and look at their fluorescence and Stokes shifts properties, once again, as a function of temperature. So here are the results. Now, these are earlier data. Often we understand things in retrospect because, in fact, we didn't have any theory to guide us, and we're trying to build a conceptual basis that will stand the test of time. So here you see now the control tryptophan far away from the active site. The energy of activation for catalysis, this is significantly larger. This is a more adiabatic hydride transfer reaction, is 13 kcals per mole. The Stokes shift has no energy of activation. For the tryptophan, that's the control near the other side, edge of the protein. The 2 trips that are in the active site have been measured either by protein fluorescence or by T-jump FRET, where we can do FRET between the tryptophan and the bound cofactor. And lo and behold, here are the energies of activation for catalysis on the top. Here's the energy of activation for the trip at the fluorescence at the trip that resides near the bound substrate, and here's the value for the Stokes shift. If we look at the other trip, which is near the cofactor, once again, either KCAT or T-jump FRET lead to the same energy of activation. There we are again. Totally different system. Let's go back to our model. So now, instead of just talking about millisecond catalysis and nanosecond Stokes shifts, we have a new time scale that we've introduced, which is the microsecond of FRET. And of course, our idea, once again, is that we have these sensitive anisotropic regions of protein which undergo these thermal quakes. The thermal quakes are essentially the movement of heat on a picosecond time scale in and out from the solvent to the active site. It's going back and forth all the time. This is not photoactivated chemistry. This is at equilibrium. This is what is so hard for people to understand. It's just heat transfer from the bath at equilibrium that depends on variations in local heat capacity of the globular and folded protein. Now, we would say that the reason that FRET and microsecond FRET and millisecond H transfer have the same energy of activation, once again, is that the probability of getting everything just right for barrier crossing and catalysis is still much more stringent than just getting the emission from the active site trip and its absorption by the neighboring nicotinamide ring of cofactor to overlap sufficiently to see FRET. So it's in the probabilities. So we've got these two separate terms which relate to thermal activation of these biological processes and the probability that the thermal activation will lead to a productive output. I mean, I'm very excited by all this. I know that these are ideas that are hard to convey, and they're new concepts in terms of how enzymes work. But it really is a physical description. It's not just saying, yes, enzymes stabilize transition state structures. So let's just try to finish up with pulling this together into a generic picture of catalysis. We came to this through tunneling reactions. The fact that tunneling reactions have different temperature dependencies for the primary chemistry and for the scaffold activation, that's not true for all of chemical reactions or enzyme reactions. So we want to move, in the end, to something which may be more general. Now, what do we read in the literature? If you read the literature and you're interested in protein function, you read protein dynamics is due to conformational sampling, right? It's all about conformational landscapes that undergo selection on binding of substrates. So we have the enzyme alone is wiggling and jiggling. The enzyme, the substrate, is wiggling and jiggling. Every now and then you get the ground state conformer which can go over the barrier more easily, and that's called conformational selection, if you will, that not all the conformers in the ground state can get over the barrier easily. Some are more productive than others. However, the conformational landscape is believed to be fully distributed throughout the protein. All these multiple conformers are believed to be easily accessible at room temperature, differing from each other by 1 or 2 kcals per mole. And this is really quite different from what I'm discussing here in terms of thermal quakes. So the way we think about it is that really we need a two-component, at a minimum, a two-component dynamical model in which we have the very prominent literature discussion of multitude of ground state conformers, only some of which are suitable for barrier crossing. But our view is what this conformational selection does, specifically for biology, is bring the ground states of the enzyme into a position that allows the full utilization of the protein-embedded and site-specific thermal conduit. So the conformational landscape is there and it's working, but it's working in conjunction with this other component, which is this site-selective thermal activation. So I will then try to convince you that this is not just true for tunneling in H-transfer biological reactions, but move into a more general picture, and that then we can finish up. And so a few years ago, with my current group of students, fortunately, even though you know my birth date, you don't know my birth year, but we are still plugging along, trying to solve this problem before I fully retire. And the fact is that this new group of people have worked so very hard to introduce a topic that involves the Temporal class of enzymes that represent well-studied, maybe 10% of all solved enzyme structures. And if you read the protein design literature, you see that the Temporal scaffold has been used for protein design. And so here you see the generic alternating helix beta sheet structure of the TIM barrel. And TIM barrels catalyze 5 out of 6 of the known enzyme chemical reactions. So it's an incredibly versatile scaffold that has evolved to catalyze a whole bunch of different chemical reactions. So our question is, if these thermal networks are pervasive in biology, within a scaffold like this, they're going to undergo coevolution with changes in the chemical reaction that's going on at the active site. And so what we did then was start to apply the tools I've just shown you for the various H-transfer enzymes, using as a prototype the murine adenosine deaminase. The woman who did it, Shuaiwa Gao, is currently in the lab. And this just shows you the front and back of the TIM barrel. This is the front looking down into where the substrate will bind. And this is the opposite end face of the protein that serves kind of as a wall in which all these active site components get structured. And what here is just to show you what the chemical reaction is. This is an enzyme in the purine salvage pathway in which this is the substrate adenosine is deaminated to form indosine, and then the ring structure can be recycled. And it's a metalloenzyme where we have a nucleophilic attack of a metal water, which is converted to a hydroxide on the purine ring, which then leads to a tetrahedral intermediate and release of ammonia and generation of the product inosine. So it replaces an NH2 by an OH group, classic organic chemistry, and it's catalyzed by a metal water in this case, assisted by a base and an acid. Now, the reason we chose this is an extremely well-studied system. It is, in fact, an enzyme that's quite important, and a compound that was developed to treat refractory leukemia. It's called a transition state analog. It's a picomolar binding inhibitor, has been analyzed with regard to its Ki, and also a crystal structure is available with the tightly bound inhibitor. And this on the right shows you the active site with the bound pentostatin inhibitor, the picomolar binding inhibitor, just to give you an idea of what the active site looks like with the 4 ligands to the metal, a histidine base, glutamate, active site acid, and then the bound transition state analog. The key feature of adenosine deaminase is that there are a wall of hydrophobic residues. Here we go again on the back face of the bound substrate. And one of these is Phe61. So there are 4 residues that keep the position of the substrate just so. So we mutated Phe61 to alanine, and we found a whole series of enzymes that show a linear trend in the energy of activation. Here we are again. We can make a single hydrophobic side chain into a mutation and change the energy of activation for catalysis. And we chose 2 of these to do the HDX spatial resolution of the thermal network, and they're shown with an arrow. One of them has an elevated energy of activation relative to wild type quite considerably, and the other has the same energy of activation, and they both show the same decrease in rate. So we can separate out an impact of mutation on the increase in the barrier for getting heat into the protein, and just the reduction in rate, which comes from a restructuring of the hydrophobic wall behind the substrate. And Shuaiwa did the HDX again. I won't go through the details. Now we have wild type Phe61 ILU and Phe61 ALA. And the main idea is that ILU does not alter the energy of activation. And so any peptide, so we're once again digesting the protein, analyzing all the peptide, any peptide that is unchanged in ILU relative to ALA, which does change the energy of activation, cannot be on the thermal network. So in this way, by comparing the mutant of interest and the control, we were able to subtract out the peptides that may be on mutation altering the rate but not the energy of activation. And we get a thermal network now that is refined, that is corrected for the impact of the mutation on the rate rather than just on the energy of activation. And this is the final thermal network that we got out. Now, what's interesting is that, once again, this is contacting solvent interfaces, and it extends across the entire face. The zinc is in here, the substrate binds here, the acid and base are here. And there is a region we picked up that involves the extended ribose ring of the substrate. But the main thermal network is going from left to right, or right to left, and extends across the entire face of the protein, and is absent from the back wall, which is holding all the active site components in place. Just to bring this up to date, that was without any ligand bound. We repeated those experiments with this tight picomolar inhibitor, pentostatin. And the two things I want to point out is that if we just look at the impact of pentostatin binding on protein, it's pretty much tightening the protein both on the front and back face. But the thermal network now that we see just comparing apoenzyme to enzyme with pentostatin. So this is the impact of mutation that allows us to infer the thermal network in the presence of pentostatin, whereas previously we had shown the thermal network in the absence of any bound ligands. So they're not identical, but they show this similar region that goes across this front face of the ball of the tin barrel. So I think that's a question that people often ask: what happens if you bind a ligand? If you're looking at all these experiments with apoprotein, how do you know it really relates to binding of ligands such as substrates or transition state like molecules? We've so far completed almost 4 of the 5 classes of tin barrels using this methodology of looking for mutations that alter the activation energy, then repeating the hydrogen-deuterium exchange. Here's the murine adenosine deaminase that I discussed in a little bit of detail. Here's a very different chemical reaction. This is a hydrolase, a lyase. We have some information on a methyltransferase and work in preparation on an oxidoreductase. In every case, there is a different chemical reaction and a different thermal network. So we believe there are reaction-specific thermal networks in response to evolving active site chemistry. So dynamics. The cumulative data indicate the role of site-specific thermal activation networks that arise in the context of the anisotropic structures of globular proteins. Biophysical probes indicate the primary importance of the scaffold. We attribute this to a combination of conformationally distributed protein landscapes with reaction-specific thermal networks. These are all rare events. These principles first emerged from studies of tunneling that are likely to be relevant to off-catalysis, and we propose a co-evolution of embedded site-selective thermal conduits along with active site structures. Everything is undergoing evolutionary pressure together. That's our model. And now we've just generalized from tunneling into chemistry of many different sorts that occurs with enzymes. All right, folks. Enough. When I was thinking about what to tell you today, I really couldn't resist, because we have come to these ideas late in my career, and they really follow from very adventitious observations early on. And I believe give us a new view into how enzymes work and take us to another level, not only of understanding, but design, and also using how we use soft materials, for example, to catalyze specific chemistries by introducing these thermal networks as a way of activating, particularly if we want to do active site-specific quantum phenomena within an embedded protein scaffold. So I've trained close to 100 students and postdocs. Sue Miller, who's on the first row, was my first graduate student. Thank you, Sue, for being here. And I indicated with an arrow the kinds of studies I talked to you today about are just part of the overall trajectory of my laboratory, but they started in the first decade with our demonstration of hydrogen tunneling. And then in the second decade, allowed us to uncover enzyme systems where we could relate the protein dynamics to the actual active site chemistry. And by the third decade, we were deep into these vibronically non-adiabatic models and the separation of coordinates for chemistry from protein scaffold activation. And I'm very proud of my group currently because we're a small group, and in a short period of time, we've been able to take these ideas from simple spatial resolution into temporal resolution and extend them into the generic area of all of enzyme catalysis. So with that, here we were in 1980. Most of you don't recognize anyone in this picture. But there was a group of biophysical chemists when I came to Cal, and I was hired into the biophysical chemistry group, even though, as I said, I consider myself more of a bioorganic or a biochemical scientist. And Bob Harris was kind of the leader of this ill-behaved group. And this is John Hurst, Rich Matthews, Ken Sauer, and Nacho Tinoco. And everyone is still alive except for Nacho, who died in 2016. And this biophysical group has kind of faded out as chemical biology has taken hold and a whole different direction has occurred in chemistry. But there is a huge historical and intellectual core to this department that is at the interface of physics with chemistry. And in fact, I learned everything I needed to know in the beginning about tunneling from Bill Bialik, who is a well-known scientist now at Princeton, who sat us down and taught us about quantum effects and tunneling in particular. So I'm very indebted to having been hired into this group, and apologize for my lack of interest in physics for a long time, but that has changed. And I wanted to just show you some pictures because I didn't know where to start. There are so many pictures, and I figured I'd start with the millennium because otherwise we would be here even longer. And this is the lab in 2000. I was chair at the same time, and this is a picnic with the staff. This goes way back, but it's quite memorable to me. This was a reunion in 2011. And this is the group pre-COVID, and during COVID, where a small group of us gathered. And finally, I wanted to show you a picture of myself with Mildred Cohen. This was to deliver the Mildred Cohen lecture at the University of Pennsylvania. And I think Mildred was probably younger than I am now, so that was quite a while ago. And thank you very much for this opportunity to try to integrate the early history of my lab at Cal with where I think the field of enzymology is going. Thank you.
Thank you, Judith. That was incredible. We have to watch the clock a bit because there is another group that will be coming in at 12:30. So, but we do have time for a couple of questions, I think.
Yeah. By the way, this is a work in progress because this is, I welcome all criticisms and comments at this point, especially criticisms. This is what we want to hear. Yes? Yeah, I mean, honestly, at the moment, my answer to you is I really don't have a clue how we would do that. I think that these two parameters are behind the other door of entropy and enthalpy that we learn in traditional kinetic formulations. So you could say we get entropy and enthalpy from the temperature dependence, right, of reactions. But we're interpreting these enthalpies in a very different way from small molecule reactivity. We're interpreting them in the context of the larger protein structure and how heat must flow through the protein in order to activate chemistry that's happening at a remote site. So the probability is very complicated. The probability involves the conformational landscape as well, which is likely dominated by entropy and not enthalpy if the difference among all of those multiple states in the ground state is small. And then you have, on top of that, the probability that the heat flow will actually lead to those unique and magical transformations that get you over the barrier, femtoseconds, with rate accelerations of up to 10 to the 30-fold. So I don't know, Birgit, you had your hand up, yeah. I welcome suggestions at this point. Yes? Yeah. So, theoretically, this is a very active field. Experimentally, we are experimentalists. So the types of vibrations so this cone that I showed you first for SLO has been modeled, and the type of thermal excitation that's observed is in the range of maybe 30 to 100 wave numbers, and it's widely distributed throughout the whole cone, so that it looks like it's some kind of condensate, if you will, of highly coupled low-frequency modes. Now, I'm very reluctant to use words that are going to be a red flag for physicists and physical chemists because, you know, these are still considered at equilibrium, but it's the extreme coupling of all these modes. And so, yes, you can actually try to look at the frequencies, and you see, and the question is, do you deal with it harmonically or anharmonically? And I think the important treatment is the anharmonic model because of, in fact, the low frequencies. But we do have some collaborations going on where the mutants are being compared to the wild type. There is a computation with the wild type enzyme alone, but we don't have any information of the impact of mutants on what happens to these coupled. I'm having trouble here. They're not all that. I mean, at the moment, they're large. They're just various force field calculations. We really need extensive molecular dynamics simulations on these particular systems that will allow atomistic understanding. Ideally, we'd also be able to incorporate what are called out of equilibrium molecular dynamics tools that are being developed as well. So I think there's, with the identification, so now, of course, thank you for reminding me. Often when I give this talk, I plead with the theoreticians in the audience to take some interest. Given these spatially resolved regions of proteins, what can we find out about them through computation? Now, there's a lot of interest in local heat capacity in proteins, but I do think the real dynamical activation is the dynamical activation, not the thermodynamic properties, is where we want to go.
So there's a very nice symmetry having the Lewis lecturer of 2021 asking questions of the Lewis lecturer of 2022. But so as to avoid a massive traffic jam on our way out, I think we'll leave it here for now. You know where to find Judith, and I'm sure she'll be happy to receive questions later after the fact. So, but before we let everybody go, just want to mention that we will be making a small deposit into one of your research accounts that will help you continue.
We actually can use it.
And I'd also like to just present you with a very special gift. This is a piece of history from the College of Chemistry.
Mm-hmm. Yeah. I think I know what it is, having been here for 4 decades. But this is my own. Thank you very much.
So we give this only to very special people.
Yes. I love it, everyone. Oh, for God's sake, really? Is it contaminated with? Oh, is it?
Used, the bench was, that is, by a fellow by the name of Glenn Seaborg, when he was a researcher.
Now I'm really worried, really. I mean,